Okay, so amino acid oxidation, or we can also say amino acid catabolism. At the same time, we are also going to learn a particular cycle which is known as urea cycle. Okay, now, we have already learned that glucose is the actually excellent fuel, and living organisms prefer glucose. And another one, fatty acid, also if the glucose is not available, then obviously the living organism will utilize the fuel generated from fatty acid. Now, in this case of amino acids, actually that depends on the type of the organism and the metabolic condition, when or which organism will utilize the energy generated from amino acids. So obviously we know that, if you remember, according to the what type of meal we have, or what type of food we consume, there are different types of animals specifically, such as one type is called carnivores. Carnivores, they actually consume meat. So that means their most of the energy, which is 90%, is coming from amino acids, because their food is meat. Then herbivores are one. They have actually food. They get food from the plants. So actually, very small portion of the energy they get from the amino acid. Otherwise, they will get it from the glucose. And if I ask you, what is the situation of human? What are we?
We are omnivores.
Yes. So we can get the energy from amino acid as well. Okay, so that means we get the energy from like we are getting the food from meat as well as plant. So we are omnivores. If we talk about microorganisms, actually microorganisms are the one at the very beginning of the metabolism. If you remember, microbes, there are microbes which are able to fix the atmospheric nitrogen, and then eventually those nitrogens are used to synthesize ammonia, and from ammonia they can actually synthesize amino acids. Even some of the microbes, they able to scavenge amino acids from the environment. And if they need to utilize the amino acid as fuel, obviously that depends on the situation, metabolic condition of that microbe. In case of plants, actually plants, obviously they are the one which are synthesizing glucose. So for energy production, they don't need to use amino acids. Actually they catabolize amino acids to synthesize other metabolites, which are actually used to synthesize different biomolecules. So that is the different organisms, how they are using the amino acid as the source of energy. Okay, now because we are learning about the metabolism of animals, specifically we can say mammals. So in this regard, animals actually catabolize amino acids in different circumstances, and among those actually three are important, and those are: number one, when the protein is synthesized, and sometimes the proteins are also degraded. Those are the like cellular proteins, and those that the synthesis and degradation, that is known as protein turnover. Some of amino acids are released, and those amino acids, they can be oxidized. Okay. And another one, if we have a diet which is rich in protein, and obviously the amino acids we get from the protein, they will be used to synthesize, at first body's protein requirements will be fulfilled, and after that, if there are remaining proteins, those remaining amino acids, those amino acids then can be catabolized. Another one, if we are starving, or maybe a person is suffering from uncontrolled diabetes mellitus, And that means in that situation there is no carbohydrate, or maybe the carbohydrate is there but the person is unable to use it. During that situation, the cellular proteins, they are degraded and used as fuel. So these are the three metabolic conditions when the mammals, they can use amino acids as the source of energy. Okay, so now the catabolism of amino acid. Actually, if we look at the structure of amino acid, we already know the amino acid has an amino group, then a carboxylic group, and the R group. So we are not actually focusing on the R group here. The main two functional group, which is the amino group, another one is the carboxylic group. So this amino group, where the nitrogen atom is present. So the very first step of the amino acid catabolism is the removal of the amino group. Once the amino group is removed from the amino acid, the remaining structure of the amino acid is called a keto acid, or specifically alpha keto acid. And that is also known as the carbon skeleton of amino acid, because there is only the carbon and oxygen and hydrogen is present, which is like the fatty acid and the carbohydrate. But amino group is the one which is containing another atom, which is nitrogen. Then this alpha keto acid, that can undergo oxidation actually through the TCA cycle that we have learned, and where there will be the production of carbon dioxide and water, or the alpha keto acids, they are actually converted to 3 and 4 carbon compounds, and those 3 and 4 carbon compounds, they can be used to synthesize glucose by gluconeogenesis. Okay, so you already know gluconeogenesis is very important pathway, specifically when glucose is not available, or maybe in a condition when we are starving or fasting. So then the glucose, that is actually transported, and gluconeogenesis occurs in liver. If you remember, glucose will be transported to other organs of our body, such as brain, skeletal muscle. Okay, now here the important difference between amino acid catabolism and other nutrients, if we look at that, that is the removal of the amino group, because the amino group is present in the amino acid, which is containing the nitrogen, whereas other nutrients, glucose or the fatty acid, they don't have the nitrogen in their structure. So that is the main difference between the degradation or catabolism of amino acid and other nutrients. So at the very first step, the amino acid removes the amino group. Okay, amino group. And actually that amino group, then it will be further catabolized. So that amino group also undergoes metabolism or metabolic pathway in a different particular pathway. And the rest of the structure, which is the carbon skeleton of the amino acid, that will be oxidized, or will be converted to a 4 carbon or 3 carbon compound, which can be used to synthesize glucose. Okay, so now we will look at the summary summary of the amino acid catabolism in mammals. So we can get the amino acid from different sources, whether it is coming from intracellular protein, that means the protein which is stored within our body, or we can also get the amino acids from our diet. These amino acid, according to the catabolic very first step of catabolism, we learned that the amino group will be separated. Okay, and we will get the carbon skeleton of the amino acid, which is actually the keto acid. And the keto acid enters the citric acid cycle. We already learned the citric acid cycle. Even also we learned that the intermediates of the citric acid cycle is used to synthesize glucose. Specifically here we are seeing oxaloacetate is used to synthesize glucose by gluconeogenesis. Okay, now what happened to that amino group? The amino group can be used for biosynthesis of other nitrogen containing biomolecules such as again amino acids, nucleotides or biological amines. Or the amino group can be catabolized by a catabolic pathway which is called here, particularly which is happening in mammals, urea cycle. So here we can see amino group becoming carbamyl phosphate which enters the urea cycle, and the end product of the urea that urea is the compound which is excreted released by the cycle, that is actually a nitrogenous waste product, and this product is like toxic to our body. So we must, or our body actually excreted it through the urine. Okay, now actually we have to focus on this whole thing one by one. Okay, so the removal of the amino group, we get the carbon skeleton, and that carbon skeleton entering the citric acid cycle. So we are not focusing on the citric acid cycle that we have already learned, and also the gluconeogenesis we also learned. But here another one we have to learn what happened to the ammonia, but we are not focusing on the biosynthesis because our topic is the catabolism. So we have to learn the catabolism of the amino group, that is the urea cycle. Okay, so this is the metabolic fates of amino groups. Okay, amino groups. We are focusing on, if you look at this cycle, the amino group, not the another part where the carbon skeleton entering the TCA cycle. Okay, so this is the liver, or we can say particularly the liver cell hepatocyte, because amino group catabolism occurs in the liver. So the within the liver, there are protein as well. So the protein can be degraded, which are the cellular proteins, can be degraded into amino acids, or also we can get the amino acids from the protein, from like our dietary protein. So all of these are the amino acid. This is the general structure. You can see the amino group is shown in pink shaded color, and the very first step of the catabolism, we said that the amino group is removed. So this amino group is removed, where obviously it will go to another compound, and that compound is here. If you look at this reaction, alpha ketoglutarate. Okay, alpha ketoglutarate receives the amino group and it becomes glutamate, and the remaining structure of the amino acids, now it is becoming the alpha keto acids. Do you get it? Is it clear?
Yes, ma'am.
Okay, now we can also or amino acids from different organs or different tissues of our body can enter the liver. So the first one here we are seeing from the muscle, and from the muscle we get the amino acid alanine. Actually, alanine is formed from pyruvate. We know that in the in the muscle glycolysis occurs, and after the glycolysis pyruvate is formed, and pyruvate actually is a keto acid. Pyruvate receives the amino group from the amino acids of the muscle, and then pyruvate is converted to alanine. Now the alanine goes to the liver, and here this is the alanine. Now the amino group is transferred to the alpha ketoglutarate, which becomes glutamate, and alanine becomes pyruvate. And if you remember, the pyruvate then it can actually convert it into acetyl CoA, or maybe pyruvate can be also used to synthesize glucose. Okay, next one, again other amino acids, that means which are coming from other tissues, even from muscle, they are actually entering the liver as glutamine. Okay, so glutamine is actually collecting the amino groups from different tissues and delivering the amino group to the liver. Or we can say that actually in our body, the two amino acids, glutamate and glutamine, they are the most abundant amino acids, and their function is to collect the amino groups from the different tissues and then deliver the amino groups to the liver. So here glutamine goes to the liver. You can see this is the amide nitrogen, amide amino group. So glutamine then release that amino group. Okay, not that like functional amino group. So this is the additional amino group, which is the amide nitrogen will be released. After releasing that amino group, now the glutamine is becoming glutamate. Okay, glutamate. Okay, now here the amino group is accumulated or like stored within the liver, and now the liver catabolize the amino group, and after the catabolism different nitrogenous waste products are produced depending on the particular animal. It can be ammonia itself, it can be urea or uric acid, which are actually all of them are toxic to the animal. So all of these should be excreted from the body of the animal. So again, this is like summary of the catabolism, or we can say the collection of amino group from different sources of our body. So here is the description of that slide. Okay, now from where we are getting the amino acids, as we said, we may have it from our diet, that is the one source, another one from our storage from. So from our diet, which actually we have also learned in case of glucose as well as fatty acid. So actually amino acid or protein digestion starts in our stomach. Within the stomach, there is this gastric glands, which is like a present within the stomach lining, or we can say the epithelial lining of the stomach. And those glands are, there are different glands. One is made up of parietal cells. Parietal cells secrete HCl. Then there are chief cells. Chief cells secrete pepsinogen, and there are gastric mucosa which secretes gastrin. Okay, now actually what happens, pepsinogen, which is an inactive enzyme, it need to be activated into pepsin, and this activation requires lower pH or acidic pH. Okay, so that means the HCl is doing that. HCl is activating the pepsinogen into pepsin, and then the pepsin will start degrading the proteins into amino acids. After that, the partially digested protein that will go to the small intestine, and you can see in the small intestine the pH is 7, that is the alkaline pH, whereas the stomach pH was acidic. Now here the pH is 7 because due to the alkali which is secreted from the pancreas, as well as in the pancreas there is this exocrine cells, and exocrine cells have granules which are called zymogen granules, and eventually those granules secrete the zymogens into the small intestine, and the function of the zymogen is to activate proteases. Proteases are enzymes which degrade protein, so eventually the proteins are degraded here into amino acids, and those amino acids then they are actually absorbed in the small villi of small intestine, and the villi is made up of villus. Actually the villus contains the intestinal mucosa cells, and those mucosa they absorbs they absorb the amino acids. Okay, so by that the amino acids are absorbed and eventually they will be transported to the liver. So we get the amino acid. Now the very first step step of the catabolism of the amino acid is the removal of the amino group, and the enzyme which catalyze this reaction is called aminotransferase or transaminase. Okay, and the reaction is known as transamination reaction. So if you look at the reaction here, we can see amino acid here. L amino acid is shown because naturally occurring amino acids are L amino acid in L configuration. That's why. So amino acid here, and you can see the pink shaded amino group that is released from the amino acid, and it goes to the alpha ketoglutarate and it becomes glutamate, and the remaining structure is the alpha keto acid. And the enzyme which is doing the reaction is called amino transferase or transaminase, and the reaction is known as transamination reaction. Okay, now here also another thing we can see the enzyme requires the prosthetic group, which is the pyridoxal phosphate. Now we get the glutamate here. So the keto acid that can enter the TCA cycle. Now, what is the function of the glutamate? The glutamate actually functions as the amino acid donor. So it can donate the amino acid to the biosynthetic pathways, or maybe like amino group catabolism pathways, where ultimately the nitrogenous waste product is produced and that will be excreted from our body. So actually in the summary, the fate even in the fates of amino group, we have actually seen this reaction, but now we are seeing all of the reactions one by one. So this is the very first reaction, which is the transamination reaction or the removal of the amino group from amino acid. Okay, now second one, this reaction is happening in the hepatocytes, and here we, in the first reaction we get glutamate. We are not focusing on the keto acid now, because the keto acid enters the TCA cycle and we learned about the TCA cycle. What happened to the glutamate, that is our focus now. So this glutamate actually transported from the cytosol of the cell to the mitochondria, and glutamate undergoes oxidative deamination reaction. Okay, oxidative deamination. So oxidation is happening, at the same time amino group is removed, and the enzyme which is doing this reaction is called glutamate dehydrogenase. Obviously oxidation, dehydrogenation, dehydrogenase enzyme, and the deamination is also there. Glutamate dehydrogenase is the enzyme. So if you look at this here, the cofactor can be NAD or NADP. So glutamate release the amino group, and also this reaction requires water, and we get the alpha ketoglutarate. Okay, alpha ketoglutarate is generated. That is actually, if you go back, alpha ketoglutarate, we get glutamate. From alpha ketoglutarate, alpha ketoglutarate really accepts the amino group and becomes glutamate. And the next step, by the glutamate dehydrogenase, and the reaction is oxidative deamination, where the glutamate release the amino group and becomes alpha ketoglutarate. So the very first reaction, which is catalyzed by aminotransferase, and the second reaction is catalyzed by glutamate dehydrogenase. These two reactions together is known as trans deamination reaction. Okay, trans deamination reaction. We get the alpha ketoglutarate. So we know that alpha ketoglutarate is an intermediate of the TCA cycle. So it will enter the TCA cycle, or this can also be used to synthesize glucose. Is it clear?
Yes, ma'am.
Okay. Then next one, where we have seen that ammonia, sorry, glutamine. And glutamine is the one amino acid which is actually collecting excess ammonia and from different tissues of our body. Actually, that is collected by glutamate. And when the glutamate is receiving additional amino group, it becomes glutamine, and that reaction is catalyzed by the enzyme glutamine synthetase. And if I think, if you remember, we already learned this reaction, but we learned that from different perspective. We learned that to learn the role of ATP in a biochemical reaction. So here is the reaction: glutamate, ATP is donating the phosphate group and becomes ADP by the enzyme glutamine synthetase. At first, it give rise to glutamyl phosphate. Same reaction you learned before, glutamyl phosphate, and then comes the amino group, which actually replace the phosphoryl group, and we eventually it give rise to glutamine. Okay, so we get glutamine. This glutamine then it is actually transported through the bloodstream and goes to the liver. In the liver, this additional amino group is released in the mitochondria of the hepatocyte by the enzyme glutaminase. Okay, glutaminase. Again, in this reaction, water is required. So after the release of the amino group, now the glutamine is again converted to glutamate. And that amino group which is released, that can actually further catabolize by the amino group catabolism pathway, and it is converted to urea. Is it clear? Do you get it?
Yes, ma'am.
So at the beginning, when we actually, if we I go back this one, so this is like kind of summary, and now we are learning individually each of the reaction. So we get glutamate. Okay, now another one, alanine. We have seen that alanine is the one which is actually like coming from the muscle, and then it is delivering the amino group to the liver. So what happens in the muscle? We know muscle is actually made up of protein, and the protein is degraded, we get amino acids. The amino acids then release the amino group to the pyruvate, which are generated by the glycolysis of glucose. So pyruvate then receives the amino group and becomes the amino acid alanine by the enzyme alanine aminotransferase. Okay, alanine aminotransferase. So here again, if you look at this reaction, actually, again, the glutamate is the one which is actually donating the amino group to pyruvate, and we get alanine. At the same time, glutamate is becoming alpha ketoglutarate. Now the alanine that actually travels through the blood and goes to the liver. Within the liver, the alanine release the amino group. Again, the same enzyme, alanine aminotransferase, that amino group is accepted by the alpha ketoglutarate, which becomes glutamate, and alanine becomes pyruvate. So pyruvate actually then undergoes gluconeogenesis, and glucose is produced. The glucose is then transported through the blood to the muscle. And here the glutamate, which release the amino group to the urea cycle, where urea is produced and ultimately excreted. So this is known as glucose alanine cycle. So up to now, each of the reaction we have learned which are actually here. Is it clear to you?
Yes, ma'am.
Yes,
So what
ma'am.
is happening here? That means from where we are getting the amino groups, or what are the sources of amino groups, and how those amino groups are ultimately released in the liver. So individually, each of the reaction is discussed in the following slides, up to this slide. Okay, now we are focusing the catabolism of the amino group. Okay, now this amino group is actually ultimately after catabolism, it gives rise to different nitrogenous waste product, and that actually depends on the organism, or we can say the animal. So according to that, the aquatic species, for example, bony fishes, they actually excrete the amino nitrogen as ammonia, and for that they are known as ammonotelic. Okay, they are called ammonotelic. Next group are called terrestrial animals. Terrestrial animals, they actually need the nitrogen to be excreted, otherwise that will cause toxicity and even cause water loss. So to prevent that, they must excrete the nitrogen. That means the nitrogen which are coming from the amino acid or other nitrogen compound, and those they do by the form or in the form of urea, and because of that these terrestrial animals are called ureotelic. Ureotelic. Okay, now there are birds and reptiles which are excreting the amino nitrogen as uric acid, and that's why they are called ureocotelic. Ureocotelic. Okay, now if you look at the plants, actually plants naturally or usually they don't excrete the amino nitrogen. Rather they actually utilize it, or if they need to excrete it, that actually happen in very unusual circumstance. Okay, now because we belong to the terrestrial animals, is it? Mammals belong to which group?
Ma'am, terrestrial.
Well,
What do we understand by terrestrial animals? What do we understand by terrestrial animals?
ম্যাম
Those who
ল্যান্ডে থাকে।
okay, but why? But what about birds and reptiles? They are also live in the land. Hmm? What is the difference? Okay, birds maybe fly.
uretilic,
Hmm?
uretilic, ma'am, excreting ammonium nitrogen.
Is it? No, I'm asking about why birds and reptiles are not included into the terrestrial animals. That is my question.
ম্যাম আমরা হচ্ছে জলের কাছাকাছি, আর পার্সের হচ্ছে জল থেকে আরো উপরে।
Hmm?
মানে হিউম্যান হচ্ছে
Sorry.
জলের কাছাকাছি অবস্থান করে, আর বার্ডস এর হচ্ছে আরো উঁচু, তাই না? এরকম।
What about reptiles? Reptiles can also go to the water. Huh? Cannot. Snake can go to water. Hmm? What it is? বার্ডস তো আকাশে উঠছে, বুঝলাম সেটা একটা মেজর ডিফারেন্স আছে। রেপটাইলস, রেপটাইলস এর সাথে কি আমরা যেটা টেরেস্ট্রিয়াল এনিমালস বলছি তাদের কি মেজর ডিফারেন্স নাই? কোনো মেজর ডিফারেন্স আছে?
আচ্ছা,
হ্যাঁ,
আচ্ছা,
কি ডিফারেন্স?
আসতেছে।
আমরা তো সোজা হয়ে চলি, তাই না?
Twins.
মানে আমাদের বডিটা, আমাদের মানে টর্সো তোমার ল্যান্ড থেকে অনেক দূরে। বাট রেপটাইলস এর ক্ষেত্রে কি, স্নেক তো একদম তার বডি দিয়েই চলছে। যাদের লেগস আছে তারা হয়তোবা লেগস এর উপর ভর করে চলছে, বাট other reptiles, they actually moving based on their body. You can check it. Okay, so now we have to learn the catabolism of amino group. And in case of mammals, because we belong to the terrestrial animals, that means the amino group is converted or catabolized into urea. Okay, and that ammonia, which is actually deposited in the mitochondria of hepatocytes, That means we can say urea cycle is occurring in the mitochondria of hepatocytes. And this urea cycle is discovered by the same scientist who discovered Krebs cycle, that is the Hans Krebs in 1932. But here another scientist is also involved, and he was the medical student associate Kurt Henseleit. Okay, now the cycle obviously occurring in the liver, and from the liver, after the cycle is end, we get the nitrogenous waste product, which is urea. Urea from the liver goes to the kidney through the bloodstream, and in the kidney, it is actually then will be excreted into the urine. Okay, now the urea cycle, where it is generated from ammonia, and there are altogether 5 enzymatic reactions. So this is the urea cycle. It looks very complicated, but the above reactions we have already actually learned. Okay, so this cycle is starting in the liver mitochondria, the very first reaction which occurs in the mitochondria, but rest of the steps takes place in the cytosol of the hepatocyte. Okay, now if you look at the above, so this is the mitochondria actually, then you can see the cytosol. So those are the very first reactions where you can see amino acids. That is the reactions we have already learned. Amino acids entering the liver or hepatocyte and releasing the amino group by the transamination reaction. So here amino group, alanine from muscle, coming from the muscle, or maybe this is the amino acid which is from the liver cell itself, or glutamine which is collecting amino group from other tissues. Those are the extrahepatic tissues, and they are releasing the amino group into the hepatocyte. So we get, in this case, if you look at this reaction, the transamination reaction, after releasing the amino group, the amino acid is becoming the alpha keto acid. And here again, glutamate, if you look at this, alanine is releasing the amino group, and ultimately in the form of glutamate, that is entering the mitochondria, as well as the glutamine entering the mitochondria. Glutamine release the amino group, the amide nitrogen, if you remember, we learned already, by the enzyme glutaminase. Okay, so we get glutamate here. Glutamate then undergoes the oxidative deamination reaction, which is catalyzed by the glutamate dehydrogenase. We have already learned it, and amino group is released. Even glutamine, because when it release the amino group, the amide nitrogen containing group, that is released, and remaining will become glutamate. So this amino group now enters the urea cycle in the form of carbamyl phosphate synthase 1. So here, if you look at this reaction where the amino group condensed with bicarbonate and ATP, ATP is donating a phosphate group. And so for the involvement of ATP, the enzyme is here, carbamyl phosphate synthetase, okay, synthetase 1. And we get the compound carbamyl phosphate. So here in this structure, you can see the groups which are coming from the different substrates. This is the amino group. This carbon is coming from the bicarbonate, and the phosphate group is from the ATP. Now the carbamyl phosphate condensed with the um, starting molecule of the urea cycle, which is ornithine. Okay, ornithine. So ornithine condensed with carbamyl phosphate, and we get citrulline. That is the reaction which is occurring in the mitochondrial matrix of hepatocyte. Then citrulline goes to the cytosol of the cell. In the cytosol, citrulline actually an um, um, intermediate with um, ATP. ATP actually donating the AMP, and it gives rise to the compound or intermediate citrullyl AMP phosphate. So if you look at this reaction, the structure citrulline, citrulline's carbonyl carbon, which actually comes from the bicarbonate that interacts with the phosphoryl group of AMP. Okay, we get citrulline. Okay, now, so in the urea cycle, the first amino group enters as uh, carbonyl phosphate, and the second amino group enters the urea cycle as here aspartate. And from where we get the aspartate? So we have to go back to the mitochondrial matrix again. So here you can see the pink colored structure, oxaloacetate, and we already know oxaloacetate is the intermediate of the TCA cycle. Oxaloacetate undergoes transamination reaction by the enzyme aspartate aminotransferase. If you look at the structure of the oxaloacetate, there is this keto group which actually accepts the amino group and becomes the amino acid aspartate. Okay, the green shaded amino group. Now this aspartate goes to the cytosol and enters the TCA cycle. That means it is condensed with the citrulline. Okay, citrulline by removing the AMP. So AMP is removed, and to this carbonyl carbon, the aspartate is condensed. Okay, so if you look at the whole structure, this is actually the intermediate arginosuccinate. Okay, arginosuccinate containing the aspartate, and then aspartate amino group is here, and then the citrulline, that very fast amino group coming from the in the form of carbonyl phosphate. Okay, and the whole compound is called, or intermediate of this urea cycle is called arginosuccinate. In the next reaction, the arginosuccinate is actually cleaved or broken down into two. One is fumarate, which is the 4-carbon compound, and that mainly comes from the aspartate part, the pink part. And the pink part, if you look at, there are 4 carbons that is released as fumarate, and the remaining part is the arginine. If you remember, arginine is an amino acid. So fumarate, does it has any similarity? Do you know the compound, the fumarate? Hello, I'm asking you. Do we know fumarate?
না ম্যাম।
Yes,
Yes, ma'am.
yes, ma'am, Jani.
From where? From where? কোথায় শিখেছি ফিউমারেট?
TCS cycle.
হুম, বলো।
Ma'am, TCS cycle.
তো তোমাদের তো মানে আরো জোরে বলতে পারার উচিত, এবং অনেকেরই বলতে পারা উচিত। আমি জানি না কেন তোমরা এই জিনিসগুলো মানে বলো না, বা মানে কি তোমাদের মাঝে চলতে থাকে, আই ডোন্ট গেট ইট। আমার কাছে শুধু এটাই মনে হয় তোমরা লেখাপড়া করো না। অ্যাটলিস্ট যে টার্ম গুলো আমরা কমন বারবার পাচ্ছি, অ্যাটলিস্ট সেটা কোথা থেকে আসছে, কোথায় শিখেছি এর আগে, সেটা কি মাথায় থাকা উচিত না?
জি
উইদিন দ্য
মিস।
সেম কোর্স, জাস্ট 1 or 2 লেকচার বিফোর, সেটা কিভাবে ভুলে যাই আমরা? তার মানে তুমি পড়াশোনা করছো না। Okay, so this is the amino acid arginine. So if you look at the structure of the arginine, which is containing the amino groups, one of the amino group which is actually entering the at the beginning of the urea cycle, and the second amino group that is delivered by the amino acid aspartate. Okay, both are present. Now the arginine undergoes hydrolysis. By this reaction, the urea is released. So that urea is actually, if you look at this structure of the urea, which is containing the two amino groups and the carbonyl carbon, that at the beginning we get it in the form of carbonyl phosphate from the bicarbonate. Okay, so urea is released and the remaining structure of the arginine becomes ornithine. Ornithine, which is the starting molecule compound that is ready to enter the enter the mitochondrial matrix and ready to condense with another molecule of carbonyl phosphate. So this is the urea, and obviously this is a nitrogenous waste product. So from the liver, they will be transported to the kidney through the blood, where ultimately it will be excreted into the urine. Clear?
Yes, ma'am.
Okay. Though the cycle looks very complicated, but if you look at the starting up to the formation of amino group on the top, that you have already learned individually one by one. Here you just learning the new step where the amino group condensed with other functional group bicarbonate and ATP and give rise to the carbonyl phosphate, which then condensed with the ornithine. And another new reaction where the aspartate oxaloacetate undergoes transamination reaction and formed aspartate. That aspartate enters the urea cycle or delivers the second amino group to the urea cycle. Okay, so যদি আমরা ওইভাবে দেখি, it is not complicated. And also in this cycle, in the above there are enzymes, some of the enzymes are shown, but if you look at the cycle, actually there are no enzymes. Okay, there is no enzymes, but what type of reactions? Mainly in the step 1, that is the condensation reaction. Step 2, where ATP is like donating the AMP, which is an intermediate, that is not a stable compound, that actually functioning as the acceptor for another amino group in the form of aspartate. That is also condensation reaction, and we get arginosuccinate. Then arginosuccinate is degraded, and 4 carbon compound fumarate is released, and the remaining structure give rise to the amino acid arginine. Eventually arginine is hydrolyzed, releasing the nitrogenous waste product urea and the starting compound, which is ornithine. Okay, so at least there is not many enzymes, but at the beginning all the enzymes you have actually learned from the start of the catabolism of amino acid, how the amino groups are released from the amino acid by the very first reaction, transamination reaction, then deamination reaction, then as well as for glutamine, glutaminase reaction, we have actually learned individually. And if you understand the transamination reaction, that actually depends on the particular amino acid and the keto acid it forms. So if I ask you regarding this, what is the keto acid of aspartate? What is the keto acid of aspartate? Who can answer it?
Ma'am, oxaloacetate.
Huh?
Ma'am,
Look at this cycle.
oxaloacetate.
The slide open. Yes, oxaloacetate. So you get it then. Okay, now if I ask you, what is the keto acid of alanine? What is the keto acid of alanine?
Pyruvate.
Pyruvate.
ওকে, কারেক্ট। এটা কি আমরা বুঝে বললাম নাকি না বুঝে বললাম?
বুঝে ম্যাম।
জি ম্যাম বুঝে।
এটা কি বুঝে বলেছি নাকি না বুঝে বলেছি? ওকে, হোয়াট ইজ দ্য কিটো অ্যাসিড অফ গ্লুটামেট? What is the keto acid of glutamate?
Glutaric acid.
Ma'am, alpha ketoglutarate.
Aspartate.
Hello, can you hear me?
Yes, ma'am.
Yes, ma'am.
Sorry, I was disconnected. So my question was, what is the keto acid of glutamate?
Ma'am, alpha
Glutaric acid.
Ma'am,
glutarate.
alpha ketoglutarate.
Yes. So I hope you understood it. Is it clear how this keto acid from that amino acid happened? By what kind of reaction? What is the reaction?
Yes, ma'am.
Yes ma'am.
So what is the reaction? Tell me. What is the reaction? The amino acid is converted into a keto acid.
Dehydrogenation.
Huh? হাইড্রোজেনেশন কেমনে?
Keto.
কে বলতে পারবে? বলো।
Ma'am, condensation.
Ma'am, transamination.
ইয়েস। সো সেই ট্রান্সঅ্যামিনেশনে যদি না বোঝো তাহলে তুমি সাইকেল কেমনে বুঝবা? ট্রান্সঅ্যামিনেশন করে রিলিজ করছে, and the amino acid is converted to the respective keto acid. এই জন্য বারবার বলছি, যদি এগুলো না বোঝো তাহলে কিন্তু তুমি, আমি জানি না, বাট একটু যদি বোঝো, দে আর নট সো ডিফিকাল্ট। ইভেন আই এম নট টেলিং টু লার্ন ইউ অল দ্য structure, chemical structure, but you should understand what is happening in the reaction. এটা মুখস্থের বিষয় না। আনলেস ইউ আন্ডারস্ট্যান্ড এই অ্যামাইনো গ্রুপ, অ্যামাইনো অ্যাসিডের অ্যামাইনো গ্রুপ আছে, সেই অ্যামাইনো গ্রুপটা অ্যামাইনো অ্যাসিড থেকে চলে গেল, তাহলে যে রিমেইনিং স্ট্রাকচার থাকলো সেটা হয়ে গেল কিটো অ্যাসিড। এটা তো মুখস্থ করার কিছু আছে এখানে? আমাকে বলো এখানে মুখস্থ করার কি আছে? শুধু হয়তো বা টার্মটা মুখস্থ করতে হবে যে এটা ট্রান্সঅ্যামিনেশন রিঅ্যাকশন বলে, তাই না? এটুকু মুখস্থ করতে হইতে পারে। আর অ্যামিনো ট্রান্সফারেজ, তাও ওখানে কি, বিকজ দ্য গ্রুপ ইজ অ্যামাইনো গ্রুপ, এই জন্য। আর ট্রান্সফারেজ হচ্ছে ট্রান্সফার করছে, ট্রান্সফার। একটু বুদ্ধি খাটাও, বেসিক্স অ্যাপ্লাই করো। শুধু চোখ কান বন্ধ করে মুখস্থ করলে লাভ হবে না। ওকে, সো আমরা পেয়ে গেলাম ইউরিয়া সাইকেল। সো হিয়ার ইজ দ্য ডেসক্রিপশন। ওকে, নাও তাহলে আমরা ইউরিয়া সাইকেল দেখলাম যে একটা বাই প্রোডাক্ট তৈরি হচ্ছে ফিউমারেট। Interestingly, the urea cycle is actually connected to the citric acid cycle or TCA cycle. And this interconnected cycle is called Krebs scientist, Hans Krebs. Okay, now if you look at this structure, this is the mitochondrial matrix, and here this is the TCA cycle, and TCA cycle, from TCA cycle we get aspartate. From where we are getting aspartate, কে বলবে? TCA cycle থেকে কিভাবে আমরা aspartate পাচ্ছি, কে বলবে? I am asking you, if you understood the urea cycle and TCA cycle, you should be able to answer my question. From where we are getting aspartate? Or from which specific intermediate TCA cycle we are getting aspartate? একটু আগেই শিখেছি।
ম্যাম অক্সালো অ্যাসিডের ফরমেশন থেকে।
এত সময় লাগে কেন তোমাদের?
ম্যাম আমি
জাস্ট
ক্যাম্পাসে।
একটা স্লাইড আগে এই জিনিসটা শিখলাম, এত সময় কেন লাগে? তাহলে পেয়ে গেলাম অ্যাসপার্টেট। সো এই যে রিঅ্যাকশন কোথায় হচ্ছে, হোয়ার ইট ইজ হ্যাপেনিং? আই ওয়ান্ট টু হিয়ার ফ্রম ইউ, এই যে অ্যাসপার্টেটটা পেলাম এটা কোথায় হচ্ছে?
ম্যাম টিসিএ সাইকেলে।
হোয়াট ইজ দ্য লোকেশন?
ম্যাম মাইটোকন্ড্রিয়া।
কেন তোমরা বলতে পারো না, কেন তোমরা পার্টিসিপেট করো না, আমি বুঝি না। তোমরা যদি পার্টিসিপেট না করো না, আই এম নট গোয়িং টু গিভ ইউ দ্য অ্যাটেন্ডেন্স। সো এই অ্যাসপার্টেট পেলাম, তুমি বললে অক্সালো অ্যাসিড থেকে পেলাম। হোয়াট টাইপ অফ রিঅ্যাকশন? হোয়াট ইজ দ্য রিঅ্যাকশন?
ম্যাম ট্রান্সঅ্যামিনেশন।
Transamination. Then aspartate goes to the cytosol and enters, এটা কি সাইকেল? And enters
ইউরিয়া সাইকেল।
ইউরিয়া সাইকেলে সে কার সাথে কনডেন্স করছে?
সিট্রুলিন।
Citrulline, and give rise to arginosuccinate, which eventually breaks into fumarate and arginine. This is the fumarate, which is a 4-carbon compound. Fumarate is also another intermediate of TCA cycle, তাই না? But fumarate can actually, here fumarate is converted to malate by the enzyme fumarase. Okay, so malate যে আমরা পেলাম, তার মানে এইখানে যে এনজাইমটা কাজ করছে, সরি এটা ম্যালেট, এটা কি এনজাইম ছিল কে বলতে পারবে? ফিউমারেট থেকে ম্যালেটের হোয়াট ইজ দ্য এনজাইম? ফিউমারেজ অর হোয়াট ইট ইজ, মনে আছে কারো?
No, ma'am.
Ma'am, fumarase.
হ্যাঁ,
ম্যাম
sorry,
ফিউমারেজ।
fumarase. So fumarase is the enzyme which convert the fumarate into malate. তাহলে আমরা ম্যালেট পেলাম। এখন আমরা যদি টিসিএ সাইকেলে দেখি, টিসিএ সাইকেল also has fumarate and malate. তার মানে these enzymes, like they are present in cytosol as well as mitochondrial matrix. So once malate is produced, malate can actually go back to mitochondrial matrix to enter the TCA cycle. And এই যে কানেকশন দেখতে পাচ্ছি আমরা, অ্যাসপার্টেট ঢুকলো ইউরিয়া সাইকেলে, সেখান থেকে ফিউমারেট তৈরি হলো, we get malate, malate is again entering TCA cycle. এই যে কানেকশন or link, that is called the aspartate arginosuccinate shunt of citric acid cycle. Okay, so this is the link between these two cycles, and that is the Krebs bicycle. এখন দেখো তুমি যদি এখন এটা এই ডায়াগ্রামটার দিকে তাকাও, এটা কি সাইকেলের মতন মনে হয়? কি মনে হয়? হ্যাঁ, বাইসাইকেলের মতন মনে হয়। হ্যাঁ, so that is for fun, so it looks like, sorry, bicycle. তাহলে বুঝলাম how these two cycles are connected. এখানে ডেসক্রিপশন আছে যেটা যে বলছিলাম fumarase, this enzyme as well as malate dehydrogenase. These two enzymes are actually present in both the cytosol and mitochondria. So they are called isoenzymes. Okay, so linkage শেষ হলো। এখন আমরা দেখি about the regulation of the TCA cycle. And there are two levels by which the TCA cycle can be regulated. And the very first enzyme, and if you remember, actually, a metabolic pathway is usually regulated by the um, enzymes of the preliminary steps of the metabolic pathway. Okay, যেটা আমরা, if you remember, we also have seen that in case of uh, TCA cycle, if you remember. So in this pathway as well, the very first enzyme is the carbamyl phosphate synthetase, which actually synthesize the compound entering the urea cycle. So here, if you look at this is the reaction bicarbonate amino group and ATP. We get the carbamyl phosphate by the enzyme carbamyl phosphate synthetase 1. This enzyme, it has an allosteric regulator, and that is N-acetyl glutamate. Okay, N-acetyl glutamate. So that means it is actually activating or activator of the enzyme. So if it is present, then the enzyme is activated and the enzyme will do the reaction. In return, the N-acetyl glutamate, which is actually synthesized from the substrates acetyl CoA and glutamate by the enzyme N-acetyl glutamate synthase. Okay, synthase. So that means if this enzyme is active or the substrates of this enzyme are available, then N-acetyl glutamate will be there, and enough N-acetyl glutamate will actually ensure the synthesis of carbamyl phosphate. Again, we can see the arginine, which is also an activator of the N-acetyl glutamate synthase. So in together, we can say that N-acetyl glutamate as well as arginine, they are the activator of the urea cycle. Okay, so this is not very complicated like the regulation of TCA cycle. So this is very simple. The enzyme which is allosterically activated by its allosteric regulator, which is N-acetyl glutamate. So that means the regulator has to be present. So the substrates which are, or enzyme which are synthesizing the regulator, if they are present, the reaction will occur. Okay, now in case of mammals, we human, we actually uh, get 10 to 15% of our energy from amino acid degradation. Okay, obviously, whether the amino acid will be catabolized or not, that actually depends on the balance between the requirements of biosynthesis of the protein, and also if the amino acids are available or not. So that will decide whether the amino acid will be catabolized or not. Okay, now because we know that there are 20 amino acids, right? There are 20 amino acids. তার মানে the 20 amino acids, they are catabolized by 20 different catabolic pathways, নাকি?
Yes, ma'am.
Ma'am.
সেটাই তো হওয়া উচিত, কারণ all the amino
Yes,
acids
ma'am.
differ according to their R group. They have different R groups, তাই না? So obviously they have 20 different catabolic pathways. But interestingly, these 20 catabolic pathways converts to only 6 major products. ওই যে কনভার্জিং মেটাবলিক পাথওয়ে, তাই না, আমরা শিখেছি। সো 20টা অ্যামাইনো অ্যাসিড এসে কনভার্স করছে ইনটু 6 মেজর প্রোডাক্টস, এবং সেই 6 মেজর প্রোডাক্টস কোথায় যাচ্ছে? দে আর এন্টারিং দ্য সাইট্রিক অ্যাসিড সাইকেল। তারপরে আরো আমরা তো সাইট্রিক অ্যাসিড সাইকেলে কি শিখেছিলাম? ইফ ইউ রিমেম্বার, সাইট্রিক অ্যাসিড সাইকেল ইজ amphibolic. The intermediates of the citric acid cycle is used to synthesize other biomolecules, for example, glucose. So that means some of the amino acids, they are utilized to synthesize glucose by gluconeogenesis, whereas others are utilized to synthesize ketone bodies by ketogenesis, and maybe others are oxidized completely, that is the catabolic phase of the TCA cycle. Carbon dioxide and water are produced. এটুকু কি বুঝতে পেরেছি? Is it
Yes,
clear?
ma'am.
Yes,
Yes, ma'am.
So here is the flow chart or the summary of amino acid catabolism. So you can see actually they are divided into the two groups, glucogenic and ketogenic acids, which are catabolized and they are entering into the TCA cycle, and those TCA cycle intermediate are utilized to synthesize glucose. So they are called glucogenic, whereas other amino acids, they are mainly utilized to synthesize ketone bodies. But if you look at this figure, so these are the amino acids and they are entering TCA cycle as alpha ketoglutarate, succinyl CoA, fumarate, here oxaloacetate, pyruvate. Okay, so mainly these are glucogenic because they are directly uh, producing the intermediates of the TCA cycle. Whereas if you look at the other amino acids which are called ketogenic, so they are mainly catabolized to the 2-carbon compound acetyl CoA, or here another one acetoacetyl CoA. So that is actually one of the compound of the ketone bodies, or acetyl CoA can be further degraded into acetyl CoA. So acetyl CoA can go to enter the TCA cycle, we know that already, or maybe acetyl CoA can be used to synthesize ketone bodies. ওকে, নাও তাহলে এখন আমাদের যদি এখানে আমরা সামারির দিকে তাকাই তাহলে আমাদের কি শেখা উচিত বলো? এই যে গ্রুপ অফ অ্যামাইনো অ্যাসিডস আমরা দেখছি, দে লাইক হিয়ার যদি আমরা একদম টপে তাকাই, আর্জিনিন, glutamine, histidine, proline, so they are becoming glutamate and eventually alpha ketoglutarate. তাহলে এই পাতই আমাদের শেখা উচিত না নাকি?
Yes,
হ্যালো,
ma'am.
শেখা উচিত তাই না? এখানে আইসোলিউসিন, মিথিওনিন, থ্রিওনিন, ভ্যালিন বিকামিং সাকসিনাইল কোয়ে, ফিনাইল অ্যালানিন, টাইরোসিন বিকামিং ফিউমারেট, অ্যাসপারাজিন, অ্যাসপার্টেট বিকামিং অক্সালোঅ্যাসিটেট। আমার মনে হয় এটা তো খুবই ইজি রিঅ্যাকশন। অ্যালানিন, সিস্টিন, গ্লাইসিন, these are becoming pyruvate. এগুলো তো আমাদের শেখা উচিত, বাট আমরা শিখতে পারছি না সময়ের স্বল্পতার কারণে, যেটাই হোক না কেন শিখতে পারছি না। ওকে, সো নাও I have decided your assignment will be this. Your assignment is to write about the specific catabolism of all the 20 amino acids. So here you can see, you can like divide them as this group which is shown in this slide, and how are these catabolized into this, like for example alpha ketoglutarate, succinyl CoA, fumarate, even the ketogenic amino acids. That is your assignment. Okay,
Yes, ma'am.
Yes, ma'am.
is it clear to all of you what you have to do?
Yes, ma'am.
Okay, whatever reactions are involved here, how these amino acids, they ultimately become glutamate, and also glutamate becoming alpha ketoglutarate, that reaction is very simple, or you have already learned. And this one, how these amino acids are becoming succinyl CoA, whatever, what are the specific reactions are involved, you have to elaborate them, discuss them. Okay, even like where that particular reaction is happening, what is the location of that reaction, what is the type of the reaction, what are the enzymes are involved in those specific reactions. So that is your assignment. Okay, and about the presentation and or presentation or viva. So what do you prefer?
Ma'am, presentation.
Presentation.
You prefer presentation?
Yes, ma'am.
Yes, ma'am.
Mm. Okay, so in that case, in case of presentation, please start preparing the assignment because student numbers are more. So you have to make a group. At least I think if we can make group within 8 groups, তাহলে কতজন করে হবে? 4 জন করে হতে পারে নাকি?
5
হুম?
4 জন করেই হবে হয়তো।
5 জন করে কিভাবে হবে? এটা কি 40 জনের ক্লাস নাকি?
না ম্যাম 4 জন বলেছি।
হ্যাঁ, যদি
ম্যাম
4 জন
টোটাল
করে হয়
30 হলে 32 আর কি 4 জন করে হলে 8টা গ্রুপে।
হ্যাঁ, ওই 2 জন যেটা থাকবে সেই 2 জনকে ধরো এডিশনাল 2 জন অন্য দুটো গ্রুপে ঢুকে যাবে, এরকম হবে আর কি। সো আমার তো মনে হয় না যে 30 থাকবে, হয়তোবা এখান থেকে অনেকেই বাদ পড়ে যেতে পারে। Okay, so in that case, depending on your assignment, then we will actually তখন তো আর আমরা পুরাটা অ্যাসাইনমেন্ট প্রেজেন্ট করতে বলবো না, আমার মনে হয় তখন আমরা হয়তো ডিভাইড করে দিতে পারি। সো এখানে যদি আমরা দেখি তাহলে 1 2 3 4 5 6 7, সো এখানে 7 যেটা গ্রুপটা আছে সেটাকেও যদি আমরা ফলো করি, so ultimately these 7 groups of amino acid, how are they catabolized, whether it is through the citric acid cycle or also like আমরা যেহেতু তাদেরকে বলছি গ্লুকোজেনিক, তার মানে তুমি যদি গ্লুকোজেনিক দেখাতে চাও তাহলে তাকে তখন কি করতে হবে, তোমাকে এই সাইট্রিক অ্যাসিড সাইকেল ইন্টারমিডিয়েট থেকে তোমাকে গ্লুকোজ সিনথেসিস পর্যন্ত দেখাতে হবে। বুঝাতে পেরেছি আমি? in
Yes,
case
miss.
of in case of ketogenic as well same thing. সো এখানে তুমি ক্যাটাবলিজম দেখাচ্ছ, শুধু ক্যাটাবলিজম না, তুমি এখানে অ্যাসিটাইল কোয়ে পেলা বা অ্যাসিটাইল কোয়ে পেলা, and how are they actually, they have the like direction, they can enter the TCA cycle or they can go to the synthesis of ketone bodies, as well as if they are entering ketone bodies, because sorry, entering TCA cycle, because by the TCA cycle we know that electrons will be generated, those electrons will be transported to the TCA cycle. So you have to actually go to the end, whether it is a biosynthetic pathway or it is a catabolic pathway. আমি কি বুঝাতে পেরেছি? যদি আমি ক্যাটাবলিক পাথওয়ে বলছি যখন, তখন তাহলে আমাকে দেখাতে হবে একদম এই যে ইলেকট্রন গুলো রিলিজ হলো, ইলেকট্রন গুলো চলে গেল অক্সিডেটিভ ফসফোরাইলেশন, দ্যাট ইজ দ্য ইলেকট্রন ট্রান্সপোর্ট চেইন। আবার বায়োসিনথেসিস একই সাথে আমি দেখাবো, যেখানে এই যে ইন্টারমিডিয়েটস হাউ দে আর ইউজড ইন দ্য বায়োসিনথেসিস অফ গ্লুকোজ অর বায়োসিনথেসিস অফ কিটোন বডিজ। দুটোকেই ফোকাস করতে হবে। Am I clear?
Yes, ma'am.
And in the next class, because we are going to learn the oxidative phosphorylation, so that will help you how you are going to arrange your assignment as well as presentation. But presentation যেহেতু বলছি, এখানে যেহেতু আমরা দেখছি 1 2 3 4 5 6 7, একটা তো কম হয়। একটা যদি কম হয় সেক্ষেত্রে আমরা হয়তো বা এখানে যে এই গ্রুপটাতে একটু বেশি অ্যামাইনো অ্যাসিড নাকি, অ্যালানিন, সিস্টিন, গ্লাইসিন, সেরিন, থ্রিওনিন, ট্রিপ্টোফ্যান, এখানে
Yes,
আমরা
ma'am.
দুটো ভাগ করতে পারি। ওকে, সো এটাকে যদি আমি দুটো ভাগ করি তাহলে সেই দুটো ভাগ করে 8টা গ্রুপ হবে। আমি 8টা গ্রুপকে সেইভাবে অ্যাসাইন করে দিব, বাট দ্য গ্রুপ উইল বি ডিসাইডেড বাই মি, নট বাই ইউ। Okay, but individually you have to work on your assignment about the whole thing, how all these amino acids, 20 amino acids, what is ultimately happening to them. They are catabolized or they are used to synthesize glucose or ketone bodies. Everything you, in the assignment you have to incorporate everything. Is it clear? কারো
Yes,
কোনো
ma'am.
কোশ্চেন আছে? ওকে, নেক্সট ক্লাসে আমি ইনশাল্লাহ তোমাদেরকে গ্রুপটাকে বলে দিব অ্যাজ ওয়েল অ্যাজ দ্য পার্টিকুলার অ্যামাইনো অ্যাসিড অ্যাসাইন করে দিব। ওকে, নাও আমাদেরকে তাহলে যদি আমরা ডেটটা ডিসাইড করি, বলো তোমাদের ফাইনাল এক্সাম কবে?
ফাইনাল এক্সাম তো জানুয়ারিতে।
কবে ডেট বলো?
2 তারিখ।
তোমাদের 2 তারিখে না? আমাদের এক্সাম শুরু কি এক্সাম কি শুরু হচ্ছে 30 থেকে?
ম্যাম
ডিসেম্বরের 31
আরো আগে থেকে। ম্যাম 31 তারিখ থেকে শুরু।
না, এক্সাম কি 20, সরি
ম্যাম 26 থেকে তো
26 থেকে ফাইনাল?
ম্যাম
আমাদের একটা কোর্স 27 এ পড়েছে।
তোমাদের তো 27। আচ্ছা, তাহলে সেক্ষেত্রে কি আমরা অ্যাসাইনমেন্ট সাবমিশন 21 রাখবো?
ওকে ম্যাম, দেন প্রেজেন্টেশনটা কবে হবে?
ওকে ম্যাম।
হ্যাঁ, প্রেজেন্টেশন তাহলে তোমার ওই আগের ক্লাসে হবে হয়তোবা, যদি
ওকে
ইউনিভার্সিটি ওপেন থাকে তাহলে তো আমরা ইন পার্সন করতে পারবো, আর যদি ওপেন না থাকে তাহলে তো অনলাইন করতে হবে। ঠিক আছে,
জি ম্যাম।
তাহলে আমি ক্লাসরুমে অ্যাসাইন করে দিব ডেট সহ। হ্যাঁ, আর
ম্যাম
তোমরা অ্যাসাইনমেন্ট প্রিপারেশন যেটা করবা ইসেই করবা, ওয়ার্ড ফাইল ইউজ
গ্রুপটা
করবা।
আমরা নিজেরা করি, বানাই।
নো, আই এম সরি, ইউ ক্যান নট ডু দ্যাট। সো তুমি ডিসাইড করবে না এই কারণে, ইউ শুড হ্যাভ দা, কি বলবো, নেটওয়ার্কিং স্কিল, তাই না? তোমার তো সবার সাথে নেটওয়ার্ক তৈরি করতে হবে, নট জাস্ট সাম পিপল ইউ নো। কমিউনিকেশন, তুমি কাউকে জানো না, তার সাথে তুমি কিভাবে কমিউনিকেট করবা, দ্যাট ইজ ভেরি ইম্পর্টেন্ট, অ্যান্ড এভরিওয়ান ইন দ্য গ্রুপ শুড পার্টিসিপেট। আমি যদি কমপ্লেন পাই যে ও পার্টিসিপেট করেনি, ওকে পাওয়া যাচ্ছে না, সো তখন কিন্তু মার্কিংটাও সেভাবে হবে। So this is also your, like, what you are doing. Are you working together? Are you communicating with your group member or not? That is also part of that work assessment. Okay, so আমরা আশা করি বুঝতে পেরেছি আমাদের পুরো বিষয়টা, অ্যাসাইনমেন্ট ইনক্লুডিং আওয়ার প্রেজেন্টেশন, ইট ইজ ক্লিয়ার। তাহলে কারো যদি কোনো কোশ্চেন না থাকে তাহলে আমরা এন্ড করছি এখানে। আর নেক্সট টিউসডে ইফ উই হ্যাভ ইন পার্সন ক্লাস তাহলে আমাদের কুইজ 4 হবে না? কুইজ 4 অন
Yes,
লেকচার,
ma'am.
লেকচার্স কোনটা কোনটা?